Hello everyone, my name is Praveen Kumar and I will be presenting on a clinical trial data anonymization topic today, which will be majorly focusing on risk assessment approaches that are currently used and what are the additional benefits of using some of the approaches. In today's presentation, I will be covering on some recent initiatives by different regulatories in last few years, what and how we categorize the clinical data, and to define the approach for anonymization and risk assessment. So the categorization of clinical data is needed. And under risk approaches, currently the industry is more lean towards k-anonymity. What are the challenges with k-anonymity, and what are the benefits of L diversity and T closeness? At the end, there will be few points that need to be considered while we are following or using any of these approaches. In the recent updates, in last few years, we have seen that Health Canada have published their guidance on the public release of clinical information. EMA has also published Policy 70 and updated the external guidance for the industry. US FDA has also been doing some proof of concept, and I have also put some penalties guidance related to the clinical data sharing. And again, the EMA has also made it mandatory to use their new platform for initial applications, and that will also mandate the submission of anonymized documents that are part of initial application. So all this is leading to more and more clinical trial transparency and the need of anonymization. So first, to understand why we need anonymization, let's categorize the clinical data. So the clinical data that is generally published in a structured or non-structured way can be divided into three parts. One are the direct identifiers like subject ID or investigator name or any study personnel name. Those are the direct identifiers that using that information, anyone can identify those personnels. Then quasi-identifiers are not directly able to identify the patients or study personnel, but when this information is combined with any other or other quasi-identifiers, then there is a possibility that someone can be re-identified. Then there could be third type of information, sensitive information, which may not lead to identification, but could be a potential reason for adversary to attack on the data. So the risk is that the information shared may lead to identification of subjects. And to mitigate this risk, the anonymization of clinical data is needed. So there is an example also that if there is a voter list with information in a particular region has been already in public, then that list uh, can be linked with the data that is shared in the public domain by pharmaceutical industry, and which may lead to one-to-one matching, and the subjects or patients or study personnel could be identified. Okay, moving ahead, the assessment of anonymization. When do we say that the anonymization is achieved? So there are two approaches mentioned in Policy 70. Singling out, there shall not be any possibility to single out an individual. There shall not be any link between any records uh, within the data or outside data which can link the patient information and help in identifying. And even there shall not be any possibility of inferring with some probability that this sensitive information is linked to a particular individual. So that is one approach. And in other words, it can also be said that we need to define a risk metric, how the risk will be calculated, what will be the approach, along with the risk threshold, and then do the actual risk calculation in a quantitative manner, so that we can say with the precision that the possibility of re-identifying any subject or patient from this data is below threshold. So first, coming to the k-anonymity. So the general methods to achieve the k-anonymities are suppression and generalization. So in this data of 16 subjects, we can see that the information is very unique individually, and anyone can be identified. But then we have generalized the information based on race and age. So then we are able to achieve the k anonymity with the value of k is equal to 4, and the data has been generalized to males white who are less than 50, females from the race other greater than equal to 50 age, and females in other category of race less than 50. So the minimum equivalence class size in these three equivalence classes is 4. So we can say that k anonymity of 4 has been achieved. So what are the challenges or issues with this? There can be a homogeneity attack. So in previous slide, we have seen that we have generalized the quasi-identifiers, and the identification of patient based on that is limited to a value of k. But we have not considered the sensitive information. So there is a possibility that the sensitive information within one equivalence class is identical. So for example, in this one, the female with other category less than 50, and if adversary knows that the patient that he or she is looking for is in this equivalence class, then they know for sure that the patient has heart related problem or had a heart attack in the past. And this is also dependent on the background knowledge. So if patient has the information that a particular person participated in the trial, and they have some information that they have a heart problem, then they are sure that the patient lies in this group and they have a heart attack. So homogeneity attack and the background knowledge attack is still possible even if the k anonymity is achieved based on the threshold. Then the other concept which is defined to overcome the challenges of k anonymity is L diversity, which generally focuses on sensitive information. So in the simplest of definition, we can say that L diversity is achieved if there are minimum L different values of sensitive information within each equivalence class. So if we have 3 equivalence classes, then each of it has minimum, say, 3 different values of sensitive information, then we can say that the 3 diversity is achieved. So for the same example, if we go, we have updated the generalization approach and generalized the age from less than 50 greater than equal to 50 to less than 55 and greater than equal to 55. Race is also generalized to white, Asian, and other categories. So now we see that the anonymity of k is equal to 4 is maintained, and there is a diversity in this sensitive information as well. So we have now in the group or the equivalence class of size 8, we have all different type of sensitive information. So we can see that there is a diversity of L is equal to 3 is achieved with the change in the approach. But with the L diversity, what is coming along with it is that it is addressing the issue with k anonymity, but it is not particularly modeling around the threat or adversary knowledge, and it may not be able to protect against the more knowledgeable adversaries. L diversity may not be needed or unnecessary to achieve in some cases. For example, if there is a single sensitive attribute, the study has been done on one indication and there are no adverse events and other things are captured, in a very sensitive or in more detail, and the sensitive information is not captured, in that case, the L diversity may not be needed to achieve. And larger the value of L, the more information is needed to rule out the possible values of sensitive attribute. So these are some of the key points on L diversity, but why do we need T closeness then? So still, even if we have achieved L diversity, there is a similarity attack, which is more based on, say, we have a different sensitive information, but if it is semantically same, for example, heart attack, hypertension, and cardiac arrest, all three are related to heart, and then these are semantically similar, and we can, or the adversary knows that, okay, a patient had some heart problem based on the information from that equivalence class. And skewness, that with some possibility and probability, if we talk about this size of 8, then heart attack, hypertension, and cardiac arrest is making the frequency of 7, and there is only one suicide attempt. So the adversary is sure that, okay, there is a higher possibility that if a subject or a patient is belonging to this equivalence class, that most of them will have a heart problem. So skewness attack and similarity attack is still not covered in L diversity, and this leads to the need for T closeness. So T closeness is a kind of a betterment to L diversity by decreasing the granularity of interpreted data. It also reduces the correlation between quasi-identifiers and sensitive attribute. We generally calculate a distance between distributions. What are those two distributions? One is the distribution of sensitive information in a complete data or whole data and in the equivalence class. So it reduces the distance between the distribution, these two distributions. It also protects against the homogeneity and background knowledge attacks that we have seen in the k-anonymity. And for categorical or categorical sensitive information, it takes care of the semantic closeness of the attributes as well. So basically, if you want to understand the T closeness, then say if there is Q is a distribution of sensitive information in whole table, P is distribution of sensitive attribute in the equivalence class, then we want to reduce the size or the distance between Q and P. So if adversary had say B0 belief on the sensitive attribute before looking at the published data, and B1 and B2 are the information that adversary has, or the belief, after looking at the full data and after looking at the equivalence class data, then we want to reduce the difference between B1 and B2, and it is possible only when P is close to Q. So if P and Q are same, then B1 and B2 will be same and the impact will be minimal. So for that, we have this distance, earth movers distance calculation. This is generally dependent on the probability of a particular sensitive information in whole data and in equivalence class. So if there is a, say, heart attack in whole data, so what is the possibility of appearing this sensitive information in complete data? And then what is the possibility of the same in equivalence class set? So if that distance between these two possibilities is less, then we can say that the difference between the two distributions is below threshold or minimal as needed. So what are the consequences or challenges with the T closeness? Sometimes it may not be possible to achieve the needed L diversity and T closeness for all the cases. If there is sensitive information with the one value or two values only, we may not be able to achieve the required T closeness. Loss of data utility, if we want to achieve a T closeness, recommended T closeness of 0.2, there is a possibility that some data utility will be lost. It is hard to identify based on the T closeness value that what is the knowledge gained by the adversary. It also necessitates that sensitive attributes spread in the equivalence class to be close to that in the overall table, but sometimes it may not be possible. So when we want to implement this in our actual scenario, what would we need? So if we are dealing with the individual patient data, then it is possible that we include the sensitive information from medical history and adverse event for these calculations. But if we are dealing only with the clinical documents, then it may not be easy to identify this information, extract it, and use it for this quantitative assessment. There can be multiple sensitive attributes per patient, so we have to consider that, and if there is any particular order of event occurrence. So if there is a event that has occurred in a sequence, and it could lead to sensitive information creation, then that need to be considered. And before applying these strategies, if we want to remove the sensitive, non-sensitive events, then those can be removed as well. So these are the actual calculations that we can perform based on the formulas for the distance calculation and L diversity, but the approach may vary from one project to another or on the one data set to another based on the type of sensitive information that is shared. So that was all, and my contact details, if anyone has any questions, my contact details will be there on the PHUSE app, and we can connect anytime during the conference as well as after that. Thank you. Thank you, everyone.